Good afternoon. This presentation today will describe the development of a PBPK model for efavirenz and its use as a moderate CYP3A4 inducer for drug-drug interaction predictions. As background, efavirenz is an antiretroviral medication used to treat and prevent HIV and AIDS. It's eliminated primarily by CYP2B6 and 3A4 oxidative metabolism. And due to substantial induction of both CYP3A4 and 2B6 enzyme systems with daily dosing of efavirenz, dose adjustments for many co-administered medications have been shown to be necessary. According to the FDA, strong, moderate, and weak inducers are categorized as drugs that decrease the AUC of sensitive substrates of a pathway by either greater than 80%, between 50 and 80%, and between 20 and 50%, respectively. Efavirenz, when it's dosed at 600 milligrams daily in adults, is a moderate CYP3A4 inducer. And as such, a fully validated PBPK model for efavirenz that includes the validation of the induction potentials would be useful as a standard compound for PBPK drug-drug interaction simulations. So the objectives of this study were to develop and validate a PBPK model for efavirenz in humans using PK data obtained from the literature. Additionally, we wish to validate this model as a moderate CYP3A4 inducer for DDI predictions using available induction parameters and observed PK data from DDI studies within the literature. As methods, we use GastroPlus to develop the PBPK model and run the DDI simulations. ADMET Predictor was applied to obtain in silico predictions of key physical, chemical, and biopharmaceutical properties based on the chemical structure of efavirenz, and these were used when experimental data was not found in the literature. Plasma concentrations were extracted from the literature, and the parameters for efavirenz PBPK were derived separately for absorption, distribution, and elimination, and we used the ACAT, the Advanced Compartmental Absorption and Transit Model, to describe the intestinal dissolution, absorption, and metabolism of efavirenz. PBPK physiologies, which included organ weights, volumes, and blood flows, were generated by the pair physiology module within GastroPlus. And additionally, the efavirenz tissue distribution was modeled using a perfusion-limited model for all tissues. And the KPs were predicted using the default method, the Lukakova method, within GastroPlus. So for this development, the key mechanistic assumptions that we used were that efavirenz was absorbed from the gut via passive diffusion only, and efavirenz was considered metabolized primarily by 2B6 and 3A4, and these were the only enzymes that were included in the development. Additionally, renal elimination is minimal to negligible, and therefore was not included in the current model. The induction of CYP2B6 and 3A4 by efavirenz was included in the model to predict both steady state efavirenz PK and for the drug-drug interaction simulations. Additionally, due to the low solubility of efavirenz, dissolution and absorption is highly dependent on the particle size. So as a result, and to be consistent with literature information, the mean particle size of efavirenz formulation was adjusted to 5 microns, and this was needed to fit the absorption phase and the Tmax for the evaluated profiles. 3A4 and CYP2B6 enzymes that were used for the elimination of efavirenz, literature values for the Km and Vmax based on human liver microsomes were converted to in vivo equivalents within GastroPlus and used as the starting points for optimizing the settings for the efavirenz metabolism. The Vmax values were subsequently adjusted to better match the observed efavirenz plasma concentration versus time profiles reported in the literature. for both single and multiple dose administration of efavirenz. The initial values for the EC50 and Emax for the induction were obtained from the literature. The Emax for CYP3A4 was further optimized and validated against data from DDI studies with ketoconazole and L-fentanyl. The PBPK model for ketoconazole has previously been developed and is included as a standard CYP3A4 substrate within GastroPlus. This is just a very brief flow diagram showing the overview of the steps in the model development. In the dark blue are the standard steps used for the efavirenz development, from input parameters to the developing and validating the model itself, and then the calibrating and validation of the induction parameters. And for this, we use the ketoconazole model within GastroPlus. And in order to have more confidence in the induction parameters for efavirenz, we chose to use two CYP3A4 substrates. And for this, based on literature data that was available, L-fentanyl was chosen. And we needed to develop a model for that. Using the input parameters, we developed and validated the PBPK model for L-fentanyl and used rifampin induction to validate this model. And I'll show you briefly at the end of this talk those simulations. So if we go to the results, this table provides the physicochemical properties that were included in the PBPK model for efavirenz. These are either derived experimentally or predicted or fitted values. If we look at the lower portion, the Vmax and Km parameters for CYP2B6 and 3A4 enzymes were optimized to fit the data. The Km values were taken from the literature. For CYP3A4, the Emax and EC50 were model fit, and literature values were used for the corresponding Emax EC50 values for CYP2B6. These parameters shown in this table were used for all the drug-drug interaction simulations. So initial model development for efavirenz used a single dose of efavirenz data from which the CYP2B6 Vmax was adjusted to best fit each of the individual studies. The simulation presented on the left side of the slide shows an example data set for single dose efavirenz administration of 600 milligrams and shows the CYP2B6 Vmax values. Variability in the CYP2B6 expression was accounted for the differences between these various studies for these single dose simulations. For determining the steady state scenario, autoinduction of both CYP3A4 and 2B6 mediated efavirenz clearance occurs following the repeated doses. So we needed to include autoinduction process in the simulations to assess the appropriate parameters for steady state efavirenz. And for this, we use the DDI module. And on the right-hand panel here is a simulated multiple dose efavirenz profile with the final adjusted values for the CYP2B6 Vmax from a representative clinical study presented, and shows a reasonable fit. And this is the Vmax value that was used throughout the rest of the assessments. So for calibration of the CYP3A4 induction parameters for efavirenz, the Emax and EC50 were adjusted to match the reported data for the DDI between efavirenz induction and a single 400 milligram oral dose of ketoconazole. Here, the plasma concentration for ketoconazole administered alone are shown in blue, and the corresponding concentrations after 14 days of efavirenz induction are shown in red. Good agreement is observed between the simulated, which are the lines, and the observed data points. And the utility of the model to predict efavirenz CYP3A4 induction potential was further validated for L-fentanyl. with and without 14 days of efavirenz induction. And literature studies were available for this interaction both after oral and IV administration of alfentanil. So for this, efavirenz was administered as 600 milligrams daily for 14 days, again, to achieve the enzyme induction. And in the left figure, alfentanil was administered as a single 15 microgram per kilogram IV dose. And in the right figure, alfentanil was administered as a single 43 microgram per kilogram oral dose. Again, the blue lines represent alfentanil administered alone, and the red lines represent alfentanil administered after efavirenz induction. Both figures demonstrate a good prediction of the observed impact of efavirenz induction on alfentanil PK. So the table in this slide summarizes the predicted and observed effects of efavirenz induction on both ketoconazole and alfentanil PK parameters. If we quickly look at the Cmax, for ketoconazole, we're somewhat under-predicting the impact of efavirenz induction, and for the alfentanil, the Cmax, we're somewhat over-predicting the percent reduction. So, and if you look at the AUC values, we're fairly consistent across the board for both the oral dose administrations of alfentanil and ketoconazole and the IV administration. So this table provides for us the final step in the validation of the efavirenz model as a perpetrator in drug-drug interactions for CYP3A4 induction with CYP3A4 metabolized substrates. So these last two slides just provide the model development information for alfentanil PBPK model that was used in this validation of the efavirenz model. This table lists the final values used of physical, chemical, and biopharmaceutical properties for the final model of alfentanil. And general assumptions for this model development were that alfentanil was absorbed from the gut via passive diffusion, and alfentanil was primarily metabolized by CYP3A4 with negligible renal elimination. This slide shows the alfentanil PBPK model validation, just a representative example of clinical study data for single IV and oral doses with and without rifampin induction. We use the rifampin PBPK model that is in the standards database of GastroPlus. This representative figures, These representative figures show the IV administration on the left and oral administration on the right, and demonstrate a good fit of the simulated profiles to the observed clinical data. And this provided validation for the use of alfentanil as a CYP3A4 substrate for the presented interactions. So in conclusion, we've developed a PBPK model for efavirenz, which accurately reproduces the efavirenz exposures from published single-dose and steady-state studies. DDI simulations with efavirenz as the perpetrator, with either ketoconazole or alfentanil as a victim, accurately predict the observed exposures due to the induction of CYP3A4 by efavirenz. This developed and validated PBPK model for efavirenz could be considered suitable to be used as a standard perpetrator to evaluate the impact of moderate CYP3A4 induction on 3A4 metabolized compounds. And finally, we developed and validated a PBPK model for alfentanil, which is suitable to be used as a standard victim for DDI interaction predictions with CYP3A4 inducers. Here are briefly some references if you have the need to refer back to. Thank you for your attention.